We get experimental science. We're curious, non-judgmental.
So what we're gonna do is we're gonna put them on top of a plate surface in a very, very thin layer of water, and then we're gonna just chop them right in half. The chop streamer fits perfect. Yeah, it really does this time, Gwen. Come on now. There, for all the new people coming in, this is a planarian. It is a flatworm. So we fed them on Monday. And you might ask, well, Balint, why didn't you split them on Tuesday or Wednesday? If you split them right after feeding, that undigested food goes everywhere, and they're more likely to get an infection. Bacteria will colonize, and that's what ends up killing them. So that's why we fed them on Monday, and we waited a couple of days. We'll do the same thing again this Monday. We'll check in on them. We'll see if they've regenerated enough of a mouthpiece to be able to eat. Otherwise, we'll probably feed them later on next week. And that'll be how we end up checking in on them and seeing if they're doing well or not. So right here is a fair bit of gut tissue. And that one, there's clearly still some food in its belly. We're about to chop these animals in half. When you do that, they regenerate. So they regrow. So the tail part will actually regrow a new head, a new brain, everything. And then the head part that's cut will regrow a new tail. So we're just looking at the scope now to look at our little planarians, make sure that they're looking happy before we start the chopping process. All right, y'all, so there are Jeffreys. There's one of them. Look, he is upset! This is a trigger warning for y'all. I am going to cut him in half. He is not experiencing any pain. We will not do anything on this stream that would harm any animal. This is a standard experimental technique that is done in the lab. Luckily, I've been trained to do this. The animals feel no pain. There we go. He's flipping on over. Flipping. There we go, Jeffrey. Once it's split, we are going to put him back in the dish and he'll start his regeneration. And then in a couple of days, we'll keep checking in on them and we'll make sure to see them grow and like regrow their bodies essentially live on the stream. All right, so just so y'all know, we're about to go in. And there you go. That's it. There, that the planarian's chopped. And look, as you can see, the tail is still wiggling, no problem, right? And then the head is still wiggling, no problem. This tail is going to regrow into a brand new animal. This head is going to regrow its tail. So it's not going to lose anything in terms of its biology. But there we go. You can see there's the two components as they're growing. And you can see they're not leaking tissue at the cut site. There's already cells swirling and moving in at that cut site. Here at the tail too, we'll be able to see the cells moving on in. And that's that. So they regenerate in about 7 to 10 days. We'll see eyes for that tail. And then we'll actually end up seeing the full regeneration in maybe about 2 weeks time. We'll have 2 full planarians at that point. How many times can you keep splitting them? So you can, as long as there's stem cells in your split, you can keep splitting them. And they can live for years. And that's the tail portion right here. No, these don't reproduce sexually. They reproduce in the wild by splitting their bodies. It's a size question. It's however like big they get, then they end up splitting. We have another one here. Kinda, they will die. This particular species is about 2 to 5 years in the wild. They end up dying because their stem cells eventually do give out. If you could find a way to keep those stem cells from giving out, then you might be able to call it immortal. But at this current stage, they do have an end point. All right, y'all, we're gonna split again. Look at that, clean break. Talk about being beside yourself. Whoa! That is another split point. Thank you, Andrew. They feel stimulus, which is different from pain. They're able to feel the sensation of I've interacted with something. But to them, that doesn't necessarily mean pain. Just so y'all can see, there's that tail swimming around. So we put the tail back in. There's that tail and it's swimming next to a full planarian. See, look at that little curvature right here. That little nub right there is actually really cool because that's the wound healing site, and they've already healed up that wound quite nicely to the point where this one's swimming around no problem. This side was the more recent, earlier one that we split, where we cut at. You can see it's already essentially cauterized, and the same thing is happening here at this end right here. So y'all, what we're gonna do, actually I'm sorry, I'm still mesmerized by, just watching these cells move around, knowing that this animal that's been split twice is alive, is doing fine, and is going to be regrowing its body parts. Now, you might wonder, okay, we've cut off the head, we've cut off the tail. How does it know what side to be the head and the tail? So it turns out there's different concentrations of molecules on either side. There's high concentration on one end and low of another. And what happens there, it's pretty straightforward. The high concentration end becomes the head and the low concentration end becomes the tail. What's really cool is that researchers have been able to make that molecule be high on both ends and generate a two-headed planarian where either end has a head. They can do the same thing, decrease that gene, and make a two-tailed planarian. And so that's one of the ways that we figured out how they have this body plan and structure. Like, how do they molecularly know how to grow and make that grow happen? Again, it's just by a stretch of those molecules, if that makes sense. They would be perfectly pixelated. They would possibly be. And I want to show y'all some of these like weird double-headed planarians, just because it's really cool. My wife Lita worked with planarians in the lab, and she was able to do this kind of splitting where if you split down the middle but don't finish your cut, you'll get a two-headed planarian. And it's really, really cool to be able to see something like that. There's a couple of ways to do it. We talked about doing it genetically, so you can get rid of certain genes that give it the ability to detect which is the head and which is the tail. This is one example of it, where you will have a two-headed planarian. And that is not because of cuts, but because you have messed up the genetics, and there's no longer a directionality. There's nothing telling them which direction everything should be, but rather it's just like both sides are heads because it doesn't know which side it should become. Same thing happens this one here. This one, you have your normal planarian, but they're going to cut down the center of it. And they're not going to finish their cut all the way. And because they don't finish the cut all the way, they do a T-cut. And that has a two-headed planarian now. And what ends up, you might wonder, like, what ends up happening on this front? Like, there's a two-headed planarian. Which one's, is there a dominant head? Is there one in charge? Is there not one in charge? Eventually they do split. So eventually they'll finish that split by themselves, because that's normally how they reproduce in the wild, is that they tear themselves apart. So normally they will get rid of that component of their bodies. I will say they are sensitive to a couple of different factors. In particular, they don't like bright light. So what we're going to do now, we'll go ahead and turn off the microscope lights. Just going to add a little bit more water to them. We'll turn off the microscope light, and then we'll check in on them, as we progress tonight and see just how they're doing. There's not going to be big changes in them tonight, beyond the fact that we saw wound healing taking place. And that wound healing is a really big thing. It's really important for them to do. And because they were able to do that, some of them, like this guy here, is already booking it around and zipping around. Adding polymers to tap water, there is actually a kind of water called Planarian water. It is a recipe in the lab that you can do, and that recipe will make it so it's pH'd a particular way. So it has to be, neutral pH is 7. It has to be slightly acidic, and then it's treated with a bunch of chemicals to make it like a certain level of cleanliness. What's weird is they then do better in pond water than they would do in tap water. There's something about straight tap water that they don't do well in. And I'm not sure what it is, Andrew. It must have to do with like the mineral composition or the pH. I guess thinking about it, probably in Jersey water, it's probably a little over-chlorinated, and that extra chlorination is probably what gets them, if I had to guess. You can train a planarian to do a particular behavior. For example, and I'm going to turn down the light on these guys. What we can actually do is I can turn on up the exposure. So you'll notice that they're going to be going really slow, but right now they prefer darkness, right? They are very light sensitive. Those eye cells, when they see light, they automatically try to get away from it. That's just a feature of a planarian. What you can do is you can train a planarian to not be afraid of light. And the way you do that is by food. You put food onto a side of a dish that has light on it, and eventually the planarians learn that, hey, light means food. I don't need to be scared of this light. If you then chop them in half, and then that tail that regenerates a brand new brain, you might be like, maybe this animal will not remember with its exposure, right? Because it's been cut, it has a new brain now. It turns out that it's actually still going to remember. Not as well as the one with the original brain, but it still remembers the training, and it will still prefer that light side. It will do the same thing. It will prefer actually not just the light side, but if you train it on... Planarians don't like textured we were doing an experiment in the lab where we 3D printed a dish with a bunch of textures and grooves on it, and they really didn't like that side of the dish. But if you put food on that side, they're like, fine, I'll eat on that side. And then eventually, if you put them in that dish, they all went to the curved side, the curved edge instead of the flat edge, against their usual nature because they like the idea of food. The universal motivator or the greatest trainer. And so what it suggests is that memory isn't just in neurons, but it might also be a molecule. And that is a next level kind of scientific discovery. What we still don't know is what that molecule is. And the reason is that researchers suspect that it's a very, very small molecule of RNA. And RNA is notoriously difficult to isolate if it's really small. Because our techniques for isolating this kind of molecule is dependent on getting rid of small things. Because that's how you amplify the sequences. And so if you're getting rid of small things, well, then that's less than ideal, right, for being able to see anything on this end. But yeah, y'all, those are our little planarians. We've split them, and the idea is that when we check in on them for Microscopy Monday, we should be able to see their more development happening on that front. Like we should be seeing that regenerative action taking place.